Hello and welcome to this webinar, Utilizing Novel Sample Preparation Techniques to Enhance Cryo EM Structural Analysis. I'm Megan Thomas, Multimedia Editor at DDW. Cryo EM has been adopted into drug discovery workflows by the pharmaceutical and biotechnology industries after proving itself capable of delivering atomic resolution structures for key targets, including integral membrane proteins such as GPCRs and ion channels. More recently, improvements in throughput have seen use of the technique expand within the sector. Key to this expansion has been improvements in sample preparation efficiency and reproducibility, which have enabled the investigation of ever more challenging biomolecules. In this webinar, you will learn the importance of high-throughput cryo-EM to understanding the dynamic structural behavior of many potential drug targets, how cryo-EM is increasingly able to provide information on even the most challenging biomolecules, including samples previously too small to be analyzed, and how modern automated sample preparation techniques can help overcome significant data collection challenges. We will now hear from two researchers for whom these new methods were important factors in overcoming sample-specific challenges encountered in structural studies of macromolecules with implications for human health. But first, a bit about the speakers. Professor Wei-Jen Tang from the University of Chicago uses integrated structural biology techniques such as cryo-EM to elucidate protein-drug interactions, including studies of amyloid peptide degrading metalloproteases and chemokines involved in neurodegenerative disease. He will speak about his work in overcoming common denaturation effects to determine the cryo-EM structures of multiple states of the relatively small human protein PrEP at near atomic resolution. Radhika Malik is assistant professor at the Icahn School of Medicine at Mount Sinai and investigates the structure-function relationships of replicating damaged DNA and developing drugs to tackle resulting cancers that prove resilient to conventional chemotherapy. She will present how overcoming strong preferred sample orientation resulted in the first structure of DNA polymerase theta, both in the act of DNA synthesis and without DNA. I will now pass on to Wei-Jen Tang for his presentation.
I would like to thank the organizer, particularly Paul and Michelle, to give me the opportunity to talk about our work on proteacetic mechanism to control amyloid peptide by two human metalloproteases, pre-sequence protease and the insulin-degrading enzyme. Our laboratory used integrated structural approach to probe the function of protein that are relevant for human health. In the last few years, we have been working on a family of proteases that selectively degrade amyloid peptide. So if one think about the proteome, beta strand is most common. Among those, and the beta strand can form beta sheet. That's one of the most common structure, and reversible and irreversible cross beta sheet. In other words, protein come from either homologous or heterologous protein can come together and for its normal function, such as the lens in our eye, as a pathological condition, the most famous is Alzheimer's and plaque amyloid fibril. Reviewed by Keaton and Dawson and highlight very nicely that amyloid peptide can be associated with a neurodegenerative as a non-neuropathic systematic amyloidosis. The diseases are here and the peptide involved, the number of residue, and then the structural form before it formed the amyloid fibril. Our body have three major systems to control protein homeostasis, including chaperone for assist protein folding, ubiquitination, and the proteasome system to degrade the protein, as an autophagy whole cell to engulf and digest damaged protein. Loss of the proteostasis is one of the 9 hallmarks of aging, and for the unfolded protein to aggregate, there is a number of proteases that involved in preventing such aggregate formation. If one thinks about the protein folding landscape, you find the amyloid fibril, its cross beta sheet, is quite stable compared to the rest of the folding state, suggesting this is very hard to denature. But prior to the formation, the C formation as well as the elongation are the problem for the final product. So nature have evolved to a mechanism to control the monomer so that it's showing up, so the C cannot form, as well as it cannot effectively grow further by number of proteases. Insulin-degrading enzyme, IDE, pre-sequence protease, PrEP, are two of such proteases that selectively recognize monomeric, so the of amilyl peptide, so to prevent nucleation, prevent fiber formation. Now, first focus on pre-sequence protease, which is usually expressed in all tissue and is specific in reside in matrix of mitochondria. This nucleic protein and using the pre-sequence to target this protein into the mitochondria. This protein is specific recognized pre-sequence that is required to get about 1,000 nuclear protein into mitochondria and selectively degrade that so it don't cause problem because this kind of peptide highly positive charge as well as hydrophobic. The protein is 107 kilodalton. John King has first reported in my lab has first solved the closed structure of pre-sequence protease. It's actually in a closed state, it forms a chamber large enough to engulf about 70 amino acid long peptide. If you look at the inner chamber of this catalytic chamber, and you'll find it's highly negative charge, so can accommodate, can recognize a positive charge peptide. There are a number of hydrophobic pockets on the side away from the catalytic center or the near the catalytic site that allow to recognize hydrophobic residue of the pre-sequence as well as amyloid beta. However, this closed state is so close that the peptide cannot get in, the reactive product degrade product cannot get out. So this must have a way in the open state to capture the substrate. Unfortunately, we have failed to use crystallography to solve the open state structure. So when the resolution evolution well on the swings and single particle Cryo is good enough to solve the 100 kilodalton protein number years ago, we start to try to use Cryo to solve structure. After substantial oxidation of the protein to make it stable, we put on the grid, the particle looks good, except when you do the 3D classification, we find the particle size seems to be it's only mostly half the size, even with a full length, most of them have a missing part, suggesting the protein seems to undergo substantial denaturation during the grid making process. We use a Cryo ET to probe where the protein reside. Most of the protein are at the water interface, which tend to denature protein because this hydrophobic nature of this interface. So we then hypothesize we can use the chameleon technology to reduce exposure to this L interface to solve our denaturation problem. Chameleon technology was first chameleon and the precursor is based on the following principle. Instead of taking the grid, putting the sample on the grid and using paper to blot and drop the sample, drop the grid onto the intricate the technology allows you to plot the sample while the sample is dropping onto the grid. The sample is small enough and also the grid have this nature to absorb spread, so it's called cell wicking. The entire process can be as short as 20 milliseconds. So that's about 100-fold improvement compared to conventional method. This basically shows the process. And this shows the prototype spotted tongue prior the improvement of the technology by SPT Labtech, which substantially improved this methodology to be for general use. I highly recommend people to think about using that to take advantage of this technology. The idea is you drop it, you have a robot plunging as well as your nanowire to spread the sample. With this technology, we find all our pre-sequence protease particles are full length. This allows us to do either the apo state or substrate bound state at near atomic resolution. This is a comparison of this structure open, partially open, which is mostly closed, and that is not as close as when the substrate is bound. This slide shows the kinetic cycle of this enzyme. Enzyme can be in an open and partial open state in the absence of the substrate. Substrate come, close, and unfolding, catalysis, come back to the cycle. This slide show a simple move how the enzyme undergo large conformation change to close, the small change between the PO state and the large opening. We now have much better idea about this process using ND simulation. This gives us two major take-home message from analysis of this structure. First, we identify three regions that undergo major conformational change. We define as switch A, B, and C. A and C are cluster. A have the region that coordinate catalytic metal. This is crucial for catalysis. In the absence of the substrate, the switch region is less ordered because it has high B factor, low resolution compared to regular molecule. That's not true when the substrate bound with A beta. This is substantially improved. This is supported by the solution-based experiment hydrogen deuterium exchange by chemistry. With the substrate, you see the much less hydrogen deuterium exchange. We also find switch region that control this process, a mutation that can boost activity, suggesting this enzyme is amenable for tuning. We're hoping that we can take advantage of small molecule can tune this enzyme. This conclusion slide, our current knowledge, PrEP is undergo open, closed transition in the absence substrate. Substrate come, hydrophobic region bind switch B region, somehow trigger the close conformation that is unfolding and the cleavage and the cycle go back to open state and restart. Now let's shift the gear to talk about another enzyme that's structurally related called insulin-degrading enzyme, which was found by Malik and Icahn many years ago based on its ability to rapidly degrade insulin because insulin is a short half-life. In 80s, this enzyme was cloned and it turned out this enzyme highly conserved evolutionarily. So this enzyme cannot be specifically insulin. And it turned out it can degrade insulin as well as amyloid beta. Consequently, knockout mice develop type 2 diabetes as well as Alzheimer's disease. And the overexpression of this enzyme can reduce amyloid beta level in mice. This enzyme can degrade peptide that have opposite function like insulin and glucagon, indicating this is not selective for certain function. It's now appreciated that this enzyme can degrade many peptides that is emergenic. So we want to know how does this protein recognize emergenicity of the target peptide. We first report this dynamic structure of IDE in 2006 to show you also have sizable chamber to entrap peptide less than 80 amino acid long. You have a chamber that one side is positive, one side negative, so can entrap the peptide that have a high dipole moment. And if you look at monomer, this is N, this is C of the IDE, connect by the sign called the linker, N and C are homologous each other. And the peptide insulin in this case is entrapped inside. If you look at amyloid beta bound structure, you will find the intimate end of the peptide is from beta strand that pair with a beta sheet on the domain 2 colored in blue. The middle part also is a beta strand that form beta sheet with near at the catalytic site. This enzyme need to unfold substrate. Example is the insulin. In this case, there are two sites on the A chain showing green, four sites in the B chain showing in yellow, here the highlight in red. And because this is on the alpha helix, it cannot fit in the cloth. Therefore, it cannot be, this complete conformation cannot be degraded. And turn out we have solved the insulin, unfolded insulin and inside the catalytic chamber to show how the final product unfolding look like. We also take advantage of the spotted-on chameleon technology to solve the orientation bias of ID on the grid. That ID tend to be in one conformation, less of this orientation. Therefore, we have a low resolution. So we use spotted-on to bias and to have less bias as was the TIL series to get the complete coverage. This allows the software structure of IDE that is in open state with three possible combinations. From this structure, we find the apo state compared to the substrate bound state. Apo state catalytic regional catalytic craft have higher B factor or low resolution, either in O or PO, suggesting substrate is required to stabilize catalytic craft. This makes a lot of sense to us because this enzyme regulates immunogenicity, and immunogenic peptides tend to be highly structure flexible, so it is ready to unfold and also ready to form beta strand in such a way that can stabilize catalytic craft, versus non-immunogenic peptide cannot be ready to unfold, cannot form beta strand, so they cannot stabilize catalytic site, therefore cannot be degraded. We now have much better idea about the process, how this enzyme works, including in the open state, it takes advantage of electrostatic potential to attract the peptide into the chamber. After closing and wiggling, the IDE can unfold insulin either A chain to the side or B chain to the side in such a way that is the first cleavage on A or B, and then subsequent cleavage on the opposite chain. That leads to breakdown of insulin into N-terminal half and C-terminal half. IDE does undergo this three-state transition with preference of OPO state, which explains the allosterism of this enzyme. This is a summary slide to say that we now have a much better idea how this enzyme works. We're now taking advantage of the chameleon spotted-on technology to do the time result cryo with the hope that we can capture the initial reaction of this enzyme. And we also hope to take advantage of what we learn to control this enzyme better for the benefit of develop small molecule and that can boost activity with the hope that we can better control amyloid peptide and reduce amyloid fiber formation. This slide is a canonical slide for the funding. As particularly like to thank the collaborator Bridge Callagher and Clint Potter allow us to do the cryo analysis. And these are the people that work on this project, particularly Wenguang Liang.
Thank you. That was an excellent presentation, Wei-Jen. We'll now go across to Radhika.
Today I'm going to talk about one of the most important polymerases in translation DNA synthesis called DNA polymerase theta and how Chameleon helped drive this project. For years, the focus of our lab has been on eukaryotic replication. The replication fork is defined by dozens of proteins where these multi-subunit B family polymerases are the major players. We have DNA polymerase epsilon, which does the leading strand synthesis, DNA polymerase delta, which does the lagging strand and the leading strand synthesis, and DNA polymerase alpha, which synthesizes the Okazaki fragment. So our lab has studied all these polymerases, but a particular focus of the lab has been on these Y family polymerases shown here in gray. So whenever a DNA encounters a lesion, these multi-subunit high-fidelity polymerases get replaced by these Y family polymerases, where each of them is specialized to handle different lesions. So our lab has studied all of these polymerases, which are single subunit polymerases, except what we had trouble getting structural information on was this multi-subunit polymerase called DNA polymerase theta. For a very long time, we just had a negative stain model, but with the recent resolution revolution in Cryo EM, as well as cutting-edge sample preparation methods such as Chameleon, helped us in determining the high-resolution structure of this polymerase, and this is what I'm going to talk about today. It may come as a surprise, but our cells experience different kinds of DNA damage on a daily basis. It is estimated that there are about 100,000 DNA damaging events from various external sources such as chemical carcinogens, ionizing radiation, and UV light, as well as from internal sources such as hydrolysis and oxidation. So basically, our cells would repair these DNA damaging events by using the various DNA repair pathways, but there are some which do not have time to get repaired before the normal replication takes place, and that's where translation DNA synthesis comes in. So, a translation DNA synthesis is a central mechanism where once a high fidelity polymerase encounters a DNA lesion, as a first step, an insertive polymerase such as DNA polymerase iota would come in and insert a base opposite the lesion. As a second step, DNA polymerase zeta would come in and extend the DNA past the mismatched termini. And once DNA polymerase zeta has done its job, basically the normal replication polymerase would take over and the normal replication would continue. Due to its vital role in DNA repair and TLS, Pol zeta has emerged as a target for cancers, especially resistant to chemotherapy, such as cisplatin-based therapy. And by deciphering the high-resolution structure of this polymerase, we'll be able to look at the various protein-protein interactions which can then be targeted for drug development. So what is DNA polymerase zeta? It's a crucial enzyme that allows our cells to battle DNA damage. It belongs to a family of high-fidelity polymerases called B family polymerases, and yet is able to do translation DNA synthesis, wherein it acts as a master extender, where it extends the DNA past lesions and mismatches. In terms of the composition of POLZ, it's made up of the catalytic module Ref3, which has a polymerase domain and an inactive exonuclease domain. At its C terminus, it has cis-A and cis-B as the cysteine-rich modules. In terms of the accessory subunits, it has Pol31 and Pol32, which it actually shares with a high-fidelity polymerase, DNA polymerase delta, which also has catalytic module Pol3 with a polymerase domain and an active exonuclease domain, as well as the cysteine-rich C termini. So Pol zeta actually has a third accessory subunit called Ref7, which is unique in Pol zeta among all B family polymerases. Ref7 belongs to a family of hormoproteins which have other functions in the cell, such as acting as spindle assembly checkpoint proteins during chromosome segregation in the cell. So for a long time it was thought that there is one binding site of Ref7 in the RIR domain of Ref3. However, recently there was a second binding site discovered in human Ref3, and we were the first ones to molecularly characterize the dimeric Ref7 structure in the Pol zeta holoenzyme. So we started off with preparing the sample, and we were able to get a stoichiometric complex of the 4 subunit complex. So we started the Cryo EM studies by initially collecting a 0-degree stage tilt data, and what we were getting from that were these anisotropic preferred maps. So what we tried to do to resolve this problem was basically start tilting the stage, and in this case we tilted the stage to 40 degrees. Tilting basically increases your ice thickness as well as the motion, and then the processing and the collection both are quite tedious. And ultimately we were able to resolve the problem by getting the different views that we wanted for a successful reconstruction. However, our resolution suffered for the apoform of Pol zeta, and we were only able to achieve around 4.1 angstrom resolution. At the same time, you were looking at various sample preparation methods for the ternary complex of Paul zeta, which had the same problem of preferred orientation and was giving us these anisotropic maps. So the method that worked beautifully for us at that time was the chameleon system, which utilizes a unique technology where it basically sprays the sample on the grids. It utilizes these special nanowire grids where these nanowires basically act as blotting paper, which would quickly wick away the sample, creating an optimally thin ice for your sample. So in addition, chameleon also allows for minimal spot to plunge time, which would basically help with the preferred orientation problem because it prevents the sample from aligning to the air-water interface, as well as any protein denaturation problems if your sample has any. So the spot to plunge time that worked for us was around 130 milliseconds, and we collected the data on the chameleon grids and we processed the data using AI-based particle picker called Topaz, and using both approaches we were able to get enough side views, which we were missing before, to get a successful reconstruction. The reconstruction we got at this time was at a much higher resolution to about 3 angstroms. So this is what the structure of Paul zeta looks like. It's a ring. The DNA is in red and it contacts with the catalytic module Rev3, which is in blue. And then there are these appendages which basically come out from the core of Rev3, and they are called the Rev7 interacting region, which interacts with the two Rev7 molecules in green. We have the iron sulfur cluster at the C terminus with these two long helices, and then we have Paul 31 and 32, which complete the ring. The only subunit which does not contact with the catalytic Rev3 module is Paul 32. And if we zoom into the details, we see that a very clear density for various regions of the maps, such as the active site, the exonucleus site, as well as the iron sulfur cluster. So if we look at the catalytic module Rev3 in further detail, we find that the domains are arranged in a similar fashion as the B family polymerases. However, there are some key differences. There is this N-terminal domain, palm domain, as well as the fingers domain, which are much more elaborate in Paul zeta in comparison to the other B family polymerases. There is this unique RIR Rev7 interacting domain, which is not present in other B family polymerases. Conversely, the CTD is seen in almost all of the B family polymerases. The thumb grips the duplex portion of the DNA, and if we zoom into the active site, T0, P0 is the nascent base pair, and the fingers basically drape over them in a closed conformation. So if we look at the high resolution details of Rev3 and compare that to a high fidelity polymerase, Paul 3, which was also resolved in our lab using X-ray crystallography, we find that the active site architecture and the residues surrounding T0, P0 are actually quite similar to Paul 3, thereby telling us that the features that impose high fidelity during the nucleotide incorporation step are quite conserved and similar to a high fidelity polymerase. However, the situation at the T1 site would be quite different in case of Rev3. So T1 is a site where the lesion would come and sit in once base has been inserted by a Y family polymerase. To illustrate that further, this is the zoomed out version of the active site where the DNA is in red, the Rev3 is in cyan, and Paul 3 is in gray. If we look at the linker which connects the NTT to the palm, we see that it's actually quite closed in to the T1 site in case of Paul 3. However, in case of Rev3, it has been pulled away by this palm loop, which is a unique element in the palm domain, creating ample space for a bulky lesion to sit in at the T1 site. So basically this can be used as a structural basis for the extension reaction, which Pol Zeta is known to do so well. So now I'm going to switch gears and talk about the second important and unique subunit in the Pol Zeta holoenzyme called Ref7. As I mentioned before, Ref7 belongs to the Horma family of proteins such as MAT2, which exist as dimers. The monomers here are usually arranged in a head-to-head arrangement where basically the C helix forms a symmetric interface with the C helix of the second monomer. Whereas what we see in case of Ref7 is more of a head-to-tail arrangement where these C helices are far apart from each other. The Ref7s basically interact with the RIR region shown here in brown, and they have these conserved sequences called RBMs, which are actually conserved throughout all the species of Ref3. Ref7 dimer is in fact the organizing center of the Pol Zeta holoenzyme, where Ref7b interacts with the phosphodiesterase domain of Pol31 as well as the N-terminus of Pol32. Both Ref7s, they are interacting with the RIR domain of Ref3. And basically the Ref7 dimer is interacting with all the subunits of the Pol Zeta enzyme, and it can also serve as an adapter site for other factors to bind in the TLS machinery. Finally, we used our APO structure and compared that to our ternary complex structure. And the major difference that we found was that the fingers in the APO structure, shown here in magenta, were in an open conformation, rotated almost 15 degrees outwards in comparison to the ternary complex. And so basically what we think is that Pol Zeta's fidelity is augmented by the opening and closing of fingers at the nucleotide incorporation step. So in conclusion, what we found from the Cryo EM structures of Pol Zeta is that it has a pentameric ring-like architecture. We have proposed structural elements such as the entity palm linker and the fingers domain, which would allow this polymerase to synthesize DNA as well as to perform translation DNA synthesis. In terms of the accessory subunits, we have found a novel arrangement of the Ref7 dimer, which has never been seen in the Horma family of proteins. Pol Zeta has emerged as a drug target for chemo-resistant tumors, and for a long time the efforts to target this enzyme has been hindered by a lack of structural information on this enzyme. And now that we have this valuable information, these critical protein-protein interfaces can now be targeted for drug development. So finally, I would like to thank all the people who were part of this work, my advisor, Dr. Anil Agarwal, my collaborators in Mount Sinai, Iban, Jakob, and Riku, our collaborators in NYSPC as well as Texas, and a special thanks to Bridget, who helped develop Spotodon and Chameleon, for her valuable insights on this project. And lastly but not least, thank you Chameleon for making this project possible and giving us this beautiful high-resolution map of this critical enzyme in the DNA repair field. Thank you.
Thank you, Radhika, and thanks again to Wei-Jen for your presentations. They were highly informative. Before we go over to the questions, I'd just like to remind everyone that this webinar will be available to watch on demand at www.ddw-online.com. Radhika, Wei-Jen, and Paul, we will now go over to questions if you're ready to turn on your cameras.
I'm ready, thank you. Hello, everybody. Thanks to Radhika and Wei-Jen for the talks there, as we've said. I'm going to handle the question and answer session, at least in the round. So some of the questions that have come in, I'll cherry pick them now, or I'll pick some out of the list. First one for you, Wei-Jen. Have you characterized the particle distribution within the ice layer from chameleon grids?
Yeah.
It's a good one.
So we have done a pyramid analysis, and the distribution is similar to what we expected. In other words, we see most of denatured, half molecule, and one small fraction that is full. And the most than 95% is on the interface. Only a few is in the middle.
And perhaps related to that, an earlier question which pinged through that I just saw there. Actually, I might be able to answer this one myself, actually. So one of the earlier questions which came in during the actual talks there itself was, when freezing with chameleon, I would imagine you need higher protein concentrations. Is this the case? Now, I guess you can both answer this in a completely open way, and I'll give my view on it as well. Compared to the Vitrobot concentration, Wei-Jen, what I would say actually to couch this question is, well, we're quite open about the fact that chameleon does need a little bit higher concentration than Vitrobots, and certainly the faster you go in terms of plunge speed, then the concentration increases. We've recently published a couple of papers on this. Radhika, maybe if you want to go first this time, do you recall roughly how much more concentration you needed above your Vitrobot concentration?
Sure, Paul. So basically, we went about 3 times the Vitrobot concentration. Yeah, we tried 2 times, but that wasn't sufficient. But yeah, we had to go 3 times concentration.
Right.
Yeah. So I can give you an example of the time result Cryo EM. And so for typically the protein when we do for IDE, we use about 1 mg per ml. And when we had to do the time resolve, we had to somehow target around 3 mg per ml. In other words, when you do the mixing, we need to start with that 5 mg per ml. And then because you're mixing with a substrate, and that dropped down to around 2 to 2.5. That's where we can get our sample to behave well enough. So I do not remember we have a huge discrepancy. This I assume it's a protein dependent, and so that's what I remember. And, but to be honest, we usually get to using spotted on chameleon for our sample preparation because that's what we need to get the structure solved. So we usually don't have a luxury not to use it.
Interesting. Another question which has come in among a lot of others. I'm not sure who this one's aimed at, so I guess you can both answer this as well. How many months or years did it take to get from sample preparation problems to a final 3D reconstruction, including from any tilted data sets? So how long did each of the projects take, I guess is the question there. Obviously, someone's struggling with their own project. They need some good news.
So
come on.
I can go first here. So tilting, we started with tilting. So that took some time for us to really optimize things. The ice thickness really becomes quite, thick. The ice becomes very thick, and so the quality of your data sets are not as good. So for a year and a half, we were like struggling with our APO data set by just tilting things and finding the best grid to work with. And then chameleon came. So actually, we started when spotted on was getting developed, and then the first model of chameleon came when we got our good grids. So yeah. So it took maybe few months with chameleon. Yeah.
So I think it's all protein dependent. Some protein behave so well that data collection, you have enough data, you process, take you maybe 1 month or so, then you're on the way to look at the model and appreciate. Some will take forever. And the example like Dr. Malik, for IDE, we had to go through, optimize the sample, optimize the fab, and optimize the grip Hill series plus chameleon. That take about 2 years to even get enough good map to solve the structure. So it's complete protein dependent. If you are lucky, a month or so, once you get the sample on grade that is beautiful, and then collecting data, another 2 weeks or if your computer set up yourself, but another 2 weeks you have your map, you can enjoy your view about how the molecule look like.
Yeah, agreed. Okay, I've got some very eagle-eyed audience members today. Another question for Radhika. It appears that the 2D averages show side views in both the Vitrobot and chameleon grid. Why does the Vitrobot image produce such an anisotropic map? Have they misunderstood that, or is there a reason for that, Radhika?
So there were some side views, but we didn't have enough of them. So we had a very small proportion of the side views. So we could either go brute force and keep on collecting on our Vitrobot, or we could just, optimize our sample for chameleon and get the thing done in one collection. So that's what we tried both. I mean, we were ready to do it, but it was a very tedious, going to be a very tedious process, but chameleon worked for us. And it was able to give us enough more side views, to have a good reconstruction.
That's true for us as well, because for IDE, we also need to use a spotty tongue, and then using TIL series to enhance the one direction of view. So,
question for you, Wei-Jen, actually. It seemed quite apparent from your talk that the open states of the proteins were not really doable by crystallography. If you look at the closed state versus the open state data you have now, is there some giveaway in there about why crystallography doesn't work for the open states, or is it more that the open states are very transient in respect to the closed state?
That's an excellent question. So if you take the NB of the open state and it quickly close, particularly in prep. For IDE, that's also the same way. So closed state tend to be dominant because the nature of the temperature drop, and in solution, because you have enough, you are in the body temperature that provide enough thermal energy to flip it open. That's our view. And the linker does not provide enough hinge so that you cannot find a way to use binder to stabilize the open state.
Interesting. We take another one. Radhika, I guess another question. If we look at the Pol3 and the Rev3, they're relatively similar in broad strokes, I guess, from component, a very broad view of it. Why is it that the Rev7 is needed with Rev3, but does Pol3 have some other component of it which is equivalent to that, or is it that the Rev7 is there in the dimer to either stabilize the Rev3 more because it has this slightly bigger hole around T1 in it, or is it more to do with recruiting other partners in the process that are not needed for Pol3?
Yeah, absolutely. So actually, the second part is right. Rev7 is actually functioning as a recruiter of other elements in the TLS machinery. such as Rev1, and it actually increases the catalytic activity of Rev3. So that is something unique in Pol-Zeta in comparison to high fidelity polymerase such as Pol-3, which does not have this subunit Rev7.
Okay, thank you. One very generic question. What processing software was used, Relion or CryoSpark?
We use mostly Relion.
I used both, and I was getting very similar results from both. So I mean, they were pretty much the same.
I think there's a right and a wrong answer there, is there really, to be honest? Perhaps I can ask something a bit more generic. So what do you think the key factors were for getting to a successful sample for a Cryo EM project? What are the key things you always try to look after before you get to the vitrification process?
So you want to take first, or?
Can take first. So the sample is very important, as Wei-Jen said. The quality of your sample and the integrity of your sample is very important. I mean, during the vitrobot process, as you would know, that the sample has a tendency to get damaged. So chameleon actually, I would say, is a better approach to use. If your sample can go to that concentration where chameleon wants it to be, I think that's the best approach. But yeah, so sample is very key, and yeah, those are my inputs.
Yeah. So what we typically do is work through the as a lab from crystallography labs, we generate enough protein. So quantity of protein, don't believe you don't need a lot of protein. You do need a lot of protein to optimize your sample. So that's very important. So don't be fooled by saying, oh, you need a minimal sample. So what we typically do as solo protein, what we do is to go through a differential scan fluorometry to find out what's the temperature. We can optimize the melting temperature of the protein. So in that case, what you do is you take your sample and add a dye that will fluoresce once the hydrophobic region is exposed. Then we use additive screen, similar to crystallography. You basically change the buffer, pH, additive, know more about your protein. If ATP is a substrate that's important, you can add. That'll be good, and so forth. So that you can get through that layer. For Cryo EM, I have learned, which is counterintuitive. You actually want to run through the sample through gel filtration to get a unique peak. You take that sample directly on grid instead of trying to concentrate. Every time you concentrate, you cause problem because it causes aggregation and so forth. Where one example, if you concentrate, the protein just don't behave. So that's a common in the field. And in terms of optimize with number of the instrument, I highly encourage any institute who can afford should have a Spartan around or a chameleon around, and as well as the vitrobot. And the other, you can use conventional method and also try to use chameleon method. And you have to optimize the sample, and it's a low throughput, but that's where you can get the structure. I hope that helps.
No, I think it's good advice. Very good advice. So do you see a future then where really the way forward is to bring the vitrification process through whatever means closer to the biochemistry? I mean, you've talked about taking stuff literally off the end of a column, which is an extreme view, but so do you see a model where you literally freeze the sample straight off the column, you vitrify it, and then you use a remote microscope, for example, almost like a FedEx approach, like we've done in crystallography for many years? Do you see that becoming more prevalent?
So actually, recently what we've started doing is started using Refine. It's a mass spectrometry method to just characterize our sample, because sometimes when even if you mishandle the samples or the sample is lying for some time, you get these aggregations and things like that. So to really characterize your sample, we've been doing that now before just putting it on the grid. So that's one extra step that check step that we do now. But yeah, so we go through vitrification and then we go to a screening microscope to screen the grid.
Interesting. One late question that's just flew through the door. What strategy can we try to minimize preferred orientation problems if we don't have access to a chameleon? I should say there's increasing number of chameleons and access is growing by the week almost. So this should not be a problem for too much longer. They question things like tilt collections or whether you've used additives to maybe detergents to try and alleviate that problem.
Yeah. You're right on. So detergent would be first thing I thought about. Yeah.
Would that be Chatso or something different?
Yeah, we've tried Chapso, but I mean, it hasn't worked for us. So you have to try a bunch of detergents. I think Jenna sells a kit called V kit or something. So it has like a bunch of detergents, like 12 or something. So you can try DDM, all of that. So hasn't worked for us yet.
Same here.
I think as all scientists, I think we all appreciate that it's not straightforward, is it? Okay. I think in the interest of time, actually, what I'm going to do now is hand back to Megan. She can do the wrap up. Once again, on a personal level, thank you, Wei-Jen and Radhika, for taking the time to join us for this webinar. Two great talks and really, really nice work. So on a personal level, thank you from me. Back over to you, Megan.
Thank you for the invitation.
Thank you for the invitation. Yeah.
Thank you.
Thanks.
Thank you, everyone. And yes, to echo that, that was a really great, informative Q&A. I'm sure our audience will be very appreciative. Thanks again for your presentations. And then I'd just like to remind everyone that you can listen to this webinar again on demand at www.ddw-online.com. You can now view Drug Discovery World content online on your mobile or tablet or via the DDW app, which can be downloaded via the App Store or Google Play. Thanks for listening.
Thank you.
Bye.
Thank you. Bye, everyone.
Bye.